Good evening, ladies and gentlemen. Welcome to John Pennekamp Coral Reef State Park. Tonight we will be having a lecture for our
Can everyone on Zoom hear me? I tell you what, I feel like I'm auditioning for the Academy Awards. A little louder. I have a big mouth, so. Okay. Good evening. Welcome, everyone, to the Delicate Balance of Nature series. We're really thrilled you're here. And you may notice we've got a couple of changes tonight. I am introducing the program. I'm Ann Helmers with our board of the Friends of Pennekamp. And we are trying to make this very popular program more available to everyone. So we're going to be doing these talks the fourth Monday of every month, other than Thanksgiving, we'll do a little earlier. And we are also holding our reception. So we hope everybody got some wine, some cheese, some crackers, because we want to welcome all of our friends. If you have joined our group, you're a formal friend, an official friend. If you have not, you're still a friend, but we hope you'll sign up and become a member. So this evening, we are streaming this lecture, which is why I am mic'd up and teched up and very nervous, because I usually just ramble with no technical assistance. But before we get started with the program, I do want to say thank you first to not only Elena, our wonderful, wonderful Delicate Balance mastermind, but thank you. And can we have a round of applause for all of our volunteers? They are the people who make this possible. So I just want to let you know that we've got a few special events coming up. I want to make sure everyone knows about them, and we hope to see you all here for them. We are going to be holding the Native Plant Day this weekend, and it promises to be, as it always is, a brisk morning. So wear your sweater and come ready to get your native plants. That's a very special event here in the community. So I'm sure we'll see lots of you back for that. And our next Friends Night Out is going to be a little bit of a ringer because we're doing it during the daytime. We're going to do a flamingo painting party on February 21st from 1 o'clock to 3 o'clock here in the park. So we have a very talented artist who's going to be showing everyone how to paint flamingos. Y'all all probably know how to do that, but come and get a little bit better at it. And then our next Delicate Balance of Nature talk will be on Monday, February 23rd, and it will feature, no accident, flamingos. This is the master binds at your board working on that. So we, again, want to thank you all for coming. We're so glad that you're here. Delicate Balance is one of the most popular and longest existing programs at the park. And I want to point out that although it started back in 1992, for the last 20 years, it has been led by a pretty amazing person who's going to be your MC for the rest of the evening. So let's give it up for Elena Muratori.
Thank you, Dan. We make a good team, folks. It's been really wonderful to have the Friends join us in sponsoring this event. And yes, it was started in 1992. So this is the 35th consecutive year. And we are grateful because also the Friends helped us keep it consecutive during the pandemic, and we all did it from our homes. So we did it online with Zoom. I happen to be the Park Service Specialist for both John Pennekamp Coral Reef State Park and Dagny Johnson Key Largo Hammock Botanical State Park. I also extend my welcome. I see faces that I know from over the years and new faces, so thank you all for coming. And let's see, what else did I want to say? So I do also thank our friends for doing this pre-lecture reception, and Moat Marine for sponsoring the refreshments tonight, as well as providing a wonderful speaker. I also happen to be the volunteer manager for our two parks, and we could not run them without our volunteers. I used to say that volunteers were the icing on the cake. They are now the cake. So you have seen many volunteers here tonight doing many different jobs, and there are many more. So if any of you are interested in volunteering, please speak to me or one of the board members before you leave. You may have talents that we don't even know about, but we are looking for people to help with maintenance, with being docents, and particularly for volunteers for the Friends of Pennekamp, where you can get involved from membership records to event planning and staffing booths. There are lots of opportunities, so do talk to one of us tonight. We've provided survey cards and tickets for a drawing for a gift bag of goodies from our friends at the end of the program. I hope everyone has a ticket because that's how we count attendance and how we notice that we are meeting the requirements of the flyer marshal. Thank you in advance for taking time to fill out the survey card. I often pick speakers in the following years from the suggestions you have for topics or speakers that you've heard that you thought, this was excellent and I'd like to see them talk again or have my friends hear them speak. Let's see. Important information, just so you know, probably many of you were at the reception, but there are restrooms and a water fountain in the lobby. You will also notice if you haven't been here before that there can be a fluctuation of temperature. You may want to bring a sweater. If you didn't tonight, bring one the next time. And let's see. I also like to point out, it's very important to me that we accommodate as many people, and this is as possible. And the Florida Park Service has been committed to making our parks and programs accessible to all. All of us benefit from it. I go up the ramp, even if I don't need to. And we ask you to help us accommodate those who are chemically sensitive by not wearing scented products to these talks, if you don't mind. And we appreciate that. If you have somebody who didn't come tonight because they said, oh, I wouldn't be able to hear, or I would have difficulty with this or that. Can I bring my wheelchair? Please do call me. We definitely are wheelchair accessible, but if you have somebody who needs a sign language interpreter, give me 2 weeks advance notice and I'll try to have that happen. On to the important piece. Tonight's lecture is Beyond the Bleaching, Resilience, Restoration, and Banking on Coral's Future. Sadly, you all read that Dr. Erin Muller was going to be speaking, but she's in that winter storm and her plane didn't take off. So great though, we have her colleague, Dr. Sara Williams, who is also from Mote Marine Laboratory in Sarasota, Florida. She is the staff scientist in the Coral Health and Disease Program, and her research focuses on coral reef resistance and resilience to different stressors, why some reefs are healthier than others, what coral species and genetic individuals are more resistant to disease, and high temperatures and why, and how we can use, coral traits to predict future reef resilience. She uses innovative modeling and statistical analyses, field ecology, and experiments to advance our understanding of coral reef resilience while also working with a diverse set of collaborators and stakeholders to develop novel solutions to coral stressors like testing treatments for coral disease. Please join me in giving Sara a warm welcome.
Yes, thank you.
The passing of the crown.
All right. I think I've got this going. Thank you so much for that lovely introduction, Elena. And thank you all for having me here. I know I'm not the speaker that you thought you were going to get to see tonight, but Erin was busy with her #1 duty, which is mom duty for her daughter's gymnastics meet up in Indiana. So I like to think, let me just get this situated. Nope, we'll go there. I would like to hope that I am the next best thing to Erin speaking tonight. I say that and I realized over here that I should have grabbed a picture of myself back from 2012 when I was Erin's first National Science Foundation intern at Mote. So I've been working with Erin for over 10 years now, right? So almost 15. So I work very closely with Erin. All of our research is very similar. We are collaborators on most of our projects. I'm sorry I didn't add a picture of baby Sara in here, but here I am today. All right, so Mote Marine Laboratory, we are a independent nonprofit research institution, and we have 10 different facilities across Florida where we do research and restoration designed to really study and increase resilience in our marine ecosystems. I forgot to mention real quick, this is a mashup of Erin's slides and my slides, and I found out about this yesterday. So please bear with me if I stumble through a little bit of this, all right? And please hold all questions until the end. Thank you. Okay, so these are all of our locations in yellow where we study corals. Okay, so the Coral Health and Disease Research Program is more than just me and Dr. Erin Muller. So she leads our growing program of over 12 staff members now. We also collaborate very closely with Dr. Emily Hall's Ocean Acidification Program and Mote's Coral Reef Ecosystem Research and Restoration Programs led by Lucas Skay and Dr. Jason Spadero. Lucas doesn't have a photo on the Mote website, so I found one. Okay. All right, so all of us coral doctors really got into this because we are passionate about our marine ecosystems and we just really think coral are cool, okay? Corals are some of the coolest animals. They're my favorite animals. So I'm not gonna do a really in-depth Coral 101, but just a quick Coral 101 to make sure we're all on the same page. I see a lot of practitioners and restoration folks in the room, but just quick overview. So corals are animals. They're not rocks like a lot of people think they are at first, but they do live and build on their own skeleton. So that's the rock part of corals, right? And then corals are actually colonial animals. So each little individual circle is a coral polyp, and all those polyps are connected over their skeleton, and they live and grow and function as one coral animal. Okay. They're all like little upside down jellyfish looking things. And then if you didn't think that corals were rocks, you might have thought that they were plants, right? And that's because they look like underwater flowers. but they also do have a plant part, sort of. They have a symbiosis, a beneficial relationship with these things called zooxanthellae, or zoox for short. So they're just single cell algae that live inside their tissue. They're what give corals their beautiful colors. And they also photosynthesize, so they're creating energy from the sun, and they give most of that energy to the corals. So the corals get a bunch of energy from their zoox, and the zoox get a home and a safe place to live. So that's their beneficial relationship. And then, like all animals, corals have a very diverse microbiome. All right. So the coral host, its bacteria, its zoox, all works together to make one healthy coral animal, something that we call the coral holobiont, or whole organism. So coral reefs are vital ecosystems created by living coral animals. A healthy reef has a high abundance of living coral cover. It has high diversity, so many different species, many different types of animals living on the reef. It supports a lot of fish and other organisms. It supports our larger animals, like our charismatic megafauna that everyone loves and often forgets coral for instead. So like our turtles, our sharks. And it also just creates a lot of structure for all of its diverse inhabitants. Okay. I regret to inform you that's from St. Croix, not from Florida. So healthy reefs are extremely biodiverse. They support a quarter of all marine life. And this biodiversity creates habitat for fisheries. It provides sources of biochemical compounds for new medicines. It supports a thriving tourism economy. So more than 1 billion people benefit directly from coral reefs. And then the structure created by coral reefs also provides shoreline protection from storms. A healthy reef can reduce wave energy by up to 97%. So that's the benefits of coral reefs worldwide. Let's bring us home to Florida, okay? So Florida's coral reefs are an economic driver. They attract more than 16 million visitors per year, which supports more than 70,000 jobs for our local communities. They're worth more than $8.5 billion to our economy. And they protect our infrastructure through flood mitigation, estimated at about $823,000 per year. So they provide a massive amount of benefits to our local communities in the Florida Keys. However, we have lost over 98% of our living coral on Florida's coral reef. So although this used to be an iconic location for people to come visit and dive on a coral reef, this is now an iconic location for reef lost. Right, so this is Carey's Fort Reef from 1975 to 2014. In 1975, it was dominated by these large elkhorn corals, lots of structure. By 2014, it was a lot of rubble, right? So we've lost all of those benefits we're getting from those living corals. So our story of loss is kind of the poster child for what the rest of the world needs to prevent from happening. Okay? And this loss is due to a lot of different stressors. Kind of the two major ones I'll bring up now. So the first one is stony coral tissue loss disease, was one of the most recent diseases that has affected our coral reefs in Florida. We lost a lot of our staghorn and elkhorn coral to previous types of disease outbreaks, but now this SCTLD has gone through and it affects more than 20 species of corals, most of our massive and brain coral species, and that's really dropped a lot of our living coral cover down in recent years. So this outbreak started back in 2014. It's devastated reefs throughout the Caribbean, but it did start off of Miami and then quickly spread both north and south through Florida's coral reef. And so all of the species are affected. We're still figuring out what exactly causes the disease. We think it's part bacteria, part virus. So we're still kind of trying to understand that aspect. But what's also interesting here is that our cropper species, our staghorn and elkhorn, they're seemingly resistant to this disease. So while they got hard hit by diseases in the past, they're resistant to this one. So SCTLD, stony coral tissue loss disease, has been a major driver of loss recently, as well as coral bleaching. Now, this one you all have likely heard about sometime in the past couple of years due to the media coverage around coral bleaching on Florida's coral reef. So coral bleaching is when corals break up with their zooxanthellae because they're too stressed out, they lose their color, and their free energy source. So just to kind of walk through that process here, we start off with a healthy coral where it has its coral color, it has all of its zooxanthellae, so it's getting plenty of energy from its zoox. And it's just having a nice little happy, healthy symbiosis. Now, this is an example of a staghorn coral that's just grown on a plug. This is something that we might use in an experiment, not like we would see on our restoration trees. And if we zoom in to just one little polyp, all of these dots here are zooxanthellae. We zoom in a little closer, you can see them a little bit better, which is kind of cool. So each of those little dots is one single zooxanthellae on a healthy coral. Now, coral bleaching occurs most often when warm water temperatures stress out the corals. They get too hot and they're just suddenly like, I can't deal with these symbionts in my skin. You're gonna get kicked out. I expel you. So they kick out their symbionts and then they lose their color and look white like their underlying skeleton. And when they do that, they're losing their free energy source, right? So this is that same coral, but now bleached. Now, sometimes it's really hard to tell a bleached coral from a recently dead coral. You have to get really close up and see if you can still see their like bleached tentacles in the cups. So it's hard to see, but you will also be able to distinguish from a bleached coral with a dead coral because when they're dead for a while, the algae starts growing on it and they're very clearly gone. So luckily, corals don't always die from bleaching. They can recover. So bleaching happens because it gets hot for too long. And they can stay functioning for a while at that higher temperatures. They can withstand those higher temperatures because corals are also voracious predators. They love to eat. That's what those tentacles are for. So they can sustain themselves on the natural food source on the reef. But if it stays too hot for too long, they're just working extra hard to catch their food and they just give up, right? But if temperatures come back down to normal levels, they can take up their zooxanthellae again, get their color back, and that process usually takes a couple of weeks to maybe a couple months. So they can recover. Now, if it stays too hot for too long and the corals don't get a break, then the coral animal will die from working itself to death, basically, right? So those are the two main recent stressors that we're dealing with on Florida's coral reef. Now, although there's a very vast network of marine protected areas on Florida's coral reef, which expands from Martin County all the way at the top down to the dry tortugas, those are all these little colorful patches are different managed protected areas, right? All those areas are doing a really good job at protecting their reefs and at the scale of what they can accomplish at their local level, right? But with all those protections in place, we're still losing these corals and there's no signs of natural recovery because the issues are outside the control of our managers, right? Our issues that we're dealing with are warming ocean temperatures and disease outbreaks and other major stressors that are just kind of outside of their control. So because of that, the people like Moat and lots of other different organizations across Florida, over 15 years ago, they recognized that corals were not going to come back without a little help, right? So over the last decade, or really 2 decades now, we as a community have made an extremely efficient network of coral nurseries across the state, excuse me. So universities, nonprofits, government agencies, education groups, who else is up there? Did I miss anyone? Scouts. So all these different groups have formed a network where they are all contributing to bringing the reef back through coral restoration. So there's 14 different in-water nurseries. So these are nurseries where we keep corals and grow them out on the reef, basically using trees. So a coral tree got these nice PVC structures and the corals are hanging on it like it's leaves, right? So our branching corals love to grow there as well. That's where they like it best. And then we also have a bunch of land-based nurseries, and these land-based nurseries are where we have large tanks and can grow our corals on land, where we have more control over their environmental parameters, and they're more accessible, so we can bring in education groups a lot more easily and kind of educate everyone on what's going on with corals more easily. And we have the corals in safe holding when crap hits the fan on the reef, right? We have representatives on land, even if the ones on the reef aren't doing as well. So coral restoration has really become focused on propagation for getting these corals mass produced and put back on the reef. Now, everybody does restoration a little bit differently, but we all follow a similar cycle. I'm going to walk through this restoration cycle from what Moat does in our science-based restoration program and what that looks like in the face of climate change. So we're going to take this step by step. The first step is asexual fragmentation. So asexual fragmentation is kind of like when you take a plant cutting. So who in here has a lot of plants on their front porch like me? Right? So have you ever taken a little cutting and just to propagate it, right? And you get, oh, I have 2 plants now, which is great. Right? So we can do the same thing with corals, because remember I said earlier that corals are a bunch of little individual polyps all connected together. So as long as we have just at least one polyp, we can take another polyp off of it and it'll grow into another coral and be just fine. We just need at least one polyp to make another coral. So we can, through this cutting process, we can make more corals through fragmentation. So here in this picture, we have a rack of Orbicella faviolata, our massive starlit corals, and basically they're on these little plugs. They look like little pepperonis. So those are all little pepperoni-sized corals that have been fragmented and grown out for restoration. Now, we use a process called microfragmentation to fragment and grow out our massive coral species. There's a lot of people in this room that could talk about this way more than I could, but here I am. So microfragmentation is where we cut the corals down to a small size, about 1 centimeter squared. So about the size of your pinky nail, right? And when you cut it down to that size, you stimulate a growth response. So when you do that, they're going to grow up to 50 times faster than if they were just growing normally on the reef. So we get a lot of corals in a short amount of time. So we can grow our corals to a size that's ready for outplanting in under a year this way. So for the branching corals, however, like the elkhorn and the staghorn corals, we propagate them slightly differently. I'm going to see if I can play that video one more time. Nope. Okay. So they just grow like sticks. That's what my dad calls these corals, the sticks. We have the sticks and the rocks. So the sticks, you just take some cutters and you cut off a branch and you're able to just move them over to the next spot on the coral tree and they'll grow like that. And that's where they like to grow the best. So a little bit easier to grow them and they grow a lot faster as well. So once we make those corals, we have to grow them out a little bit before we can actually outplant them onto the reef, because when you cut them, they're a little, stressed, not happy at first. So you have to give them some time, some nurturing, some love, so they can grow out to outplanting size. So we grow them on land and in the water. Here's an overhead shot of our raceways at the Elizabeth Moore International Center for Coral Reef Research and Restoration. That's a mouthful, but that's our facility on Summerland Key. So we have these blue, large raceway tanks, and there's just racks of corals that are being grown out for outplanting purposes later on. And then we also have multiple in-water nurseries where we grow the branching and massive and bouldering corals. So we'll have our branching corals like it better underwater, but in the nurseries, but we also grow our boulder corals there as well. So while we're growing all these corals out, we can outplant more than 20,000 corals per year this way. So how do we outplant? Well, we outplant our corals in monogenic clusters. All right, that's a little bit of jargon there. So that means we're outplanting groups of the same genotype, the same genetic individual. So and we can do that because the same genotype will recognize each other and fuse. So if I'm looking out of this room, each one of us has different DNA. I don't see any identical twins. So if there are, I'm sorry that I'm missing you. But we could not cut off a finger and fuse it to our neighbor's finger, right? Because we have different DNA. But with corals, we can actually have them merge and grow together if they have the same DNA. So we outplant them in a cluster of the same genotype. And when they're all the same, they'll grow over time. Once they touch each other, they'll fuse. So they'll grow and form a larger colony even faster when we outplant them in this cluster, instead of just letting one little individual grow out over time. Okay? So here's an example of an elkhorn coral outplant, our little pepperonis over here, and then 1 year later, we are already seeing a branching colony, right? So this is how we get as many corals out on the reef as quickly as possible. So these methods have been working pretty well, right? So from 2012 to 2023, we saw about 90% survival of our outplants after 3 years on average. This gave us about 700% more living coral on the reef, which is great. We were restoring the reef, but reefs are dealing with some extreme stressors these days, especially like the summer of 2023. This summer was a real eye-opener for us, and it was very hard for many of us working in coral restoration and coral science. It was the warmest year on record for the Florida Keys, and it really was unprecedented. When I say unprecedented, it means that it was way more extreme than we expected and earlier than we expected. It just kind of came out of nowhere for us, even though we know that this is supposed to be more of a norm kind of in the future. So just to walk through this graph right here. So the blue lines up at the top, that's our sea surface temperatures through time. And then the shaded bars down here, that's a measure of heat accumulation. It's called degree heating week. So it's just how hot it was for how long. So that heat accumulation kind of builds over time in the summer. So this first part was 2022. This is pretty typical for the Florida Keys. This is 2023's. Right. This is why it was so different than what we had seen in the past, right? So it started earlier, it was more intense, temperatures got hotter than normal, and it lasted longer as well. So we really built up that heat accumulation here. Actually, they had to make a whole new alert level for coral bleaching that year because of us. That's how extreme this heat event was. So this is the depressing part of the talk. There's hope, I promise. So this picture on the left here is of our nursery with just a bunch of bleached elkhorn corals. Then we have a live elkhorn coral in August 2022 to bleached and dying in August 2023. So we saw an extreme amount of coral bleaching due to this unprecedented heat event. This event led to the functional extinction of all the elkhorn and staghorn corals on Florida's coral reef. Functional extinction. There's still some corals left. They're not totally extinct, but functional extinction means that there's so few numbers of these corals left that they're no longer fulfilling their vital roles to the ecosystem. They're no longer providing enough habitat or coastal protection, and they're also too far apart where it makes it highly, highly, highly unlikely that they're going to be able to sexually reproduce in the wild by themselves. So that's what functional extinction means. Now, this is a paper that came out in Science towards the end of the last year. We're looking at all these points here where we have observations of coral outplants or wild corals that were before and after, and we see the loss. So we see up here in the upper Keys, the red bar is the amount of mortality. So still pretty high, but there's a little bit of survival. So upper Keys, we know that their temperatures weren't quite as bad as the lower Keys, even in that extreme event. If we go down to the lower Keys, where most of our outplanting of these corals had been taking place over the last almost 2 decades, this model shows that we lost pretty much 100% of those corals. Now, this is a big picture analysis. So the statistical model that they're using to be able to combine all of these data sets together misses some of our survivors. And that's where I'm saying that there's a little bit of hope here. So there were actually survivors from this event, and we can learn from those, kind of treat this as a natural experiment. One we couldn't control, but we can still learn from. And then we'll prioritize those survivors in our efforts moving forward. Now, normally, so this is Erin's slide. I wouldn't normally put a picture of myself up here while I'm talking, but so our survivors were mostly sexual recruits. So that's the corals that we actually made through assisted sexual reproduction efforts in the lab. So here in these bar charts, we have the kind of outplant survival before the heat event. So for our Acropora palmata at different reefs, and also for our Acropora cervicornis, our staghorn coral at different reefs. And we see like, they're all 70 and 90% or so survival before the heating event. After the 2023 bleaching event, we're looking a lot worse, right? That's as the other figure said. But there's still some survival. I still have colored bars here. So we also want to look at the scales. The scale is 0 to 2% survival. This is 0 to 14. So not a lot of survival, but we did have some. And those that survived were overwhelmingly the sexual recruits of our elkhorn and staghorn corals. And so we are kind of working to understand why they were the ones that survived better. And I'll get into that a little bit more in my talk later. But another spot of hope from this bleaching event was that the massive and brain corals did much better during this whole bleaching event. Over 75% of our massive and brain corals survived the 2023 bleaching event. Now, that's really cool. These guys that grow, they grow a lot slower, which means that, it takes a little bit longer for us to produce those, but we have microfragmentation to get them to grow out. And so we can still kind of prioritize some of those species to make sure that we are outplanting and kind of saving that reef structure just by putting this kind of living barrier that'll also start accreting and making more reef. But you'll notice that this coral species is a lot flatter than our branching coral. So it's not making as much structure, which is why we still want to make sure that we incorporate our staghorn and elkhorn corals into restoration because their structure is so important. Now, we build into our restoration cycle resilience testing. So we learned that some corals survive better than others, and so we can take that and also use experiments to add in this information to tell us which corals we should be prioritizing in our restoration. Resilience testing is sort of like running a coral stress test. We do experiments to determine which coral species and genotypes, those genetic individuals, are more resistant to different stressors, because some are. So for example, in this picture, and from what we were just talking about right here, this cluster of elkhorn outplant still has its color, and it likely survived the bleaching event, where the one in the back did not, right? So we do have survivors. So before we get into how exactly we resilience test, I want to walk through, what's most important for determining resilience in corals. Now, coral health, as we talked about earlier, is supported by the entire coral holobiont, our coral host, our algal symbionts, the zoox, and the bacteria. So when they have stress responses, it's related to a breakdown in those relationships. So coral bleaching is when they break up with their zoox, right? We talked about that. And then coral disease is usually when there's issues with the coral microbiome, all right? So we have an unhappy coral holobiont. That's when we see these stress responses. And usually these relationship issues are driven by environmental changes. So the coral holobiont is really all the factors that could influence our resilience. So we have to look at the coral host, the zoox, and the bacteria. Now, like I said, we conduct experiments to identify resilient corals. So when we do this for heat tolerance, it involves putting corals in tanks and cranking the temperature up on some and keeping some at the normal control temperatures, and comparing their responses. So we'll measure their respiration, their calcification, their growth, and also like their pigmentation and how well their symbionts are doing. So if we look here at these three corals, these were three different genotypes in a heat experiment. And we have this coral on the left, which was highly susceptible. It's bleached. It's lost most of its color. We have our coral in the middle, which was moderately resistant, so it paled. But then our coral over here kept pretty much all of its color, and it's more resistant. So that's a genotype that we would identify as highly resistant and want to upscale that in our restoration efforts. Now, these experiments use a lot of biomass because we often kill quite a bit. But I try to tell my interns and my students that sometimes you have to kill a few corals to save the reef, and that's what these experiments are doing. But we're also working with collaborators to develop methods where we don't have to do those experiments that use too much biomass. So this is a project led by Dr. Kenneth Hoadley at the University of Alabama, where he's using technology and machine learning to develop non-destructive ways to assess heat tolerance among different coral genotypes. So, okay, I got it. So he's got this machine that just blasts the coral with different light frequencies, light wavelengths. So it just zaps it with light, and then it's able to measure all of these characteristics about the coral host and the symbionts that then give us a ranking of who's most tolerant to heat stress to who's least. So all that zapping. I don't need to read out. Oh, I've got this in a spreadsheet, you guys. So, we've got all that zapping leads us to kind of this metric that goes from kind of least negative to most negative. So our more positive scores are our more resistant corals, on down to our corals that would have bleached in this experiment. So we're still running a few heat exposure experiments to refine the machine learning models that work with this technology. But once we've refined those models, we should be able to just zap the corals with light and have an idea of how tolerant they are to heat and not have to run all of these experiments. So within the coral holobiont, so different coral genotypes are more resistant to heat stress, but then different symbiont types are also more resistant to heat stress. So corals can host a lot of different coral zoox species. So there's some that are more heat tolerant than others. So if we look at these outplants from April, August to October, we have a coral that is hosting a heat susceptible symbiont, and it bleaches and dies. Then we have a coral genotype that shuffles from a susceptible symbiont because it bleaches, but then it actually recovers and picks up a more tolerant symbiont type. And then we have corals that are already associating with the heat tolerant symbiont type, and they never bleach. And they just stay happy throughout. And we see this again here. I won't go into too much details, but corals that contain that heat tolerant algal symbiont can withstand about 3.6 degrees Fahrenheit higher temperatures before bleaching. So what this figure is showing is our corals with the heat susceptible symbionts are in the green. So they're just more at risk. They start bleaching earlier and dying earlier. They can't handle as much heat stress. Whereas those with the Durstidium, our heat tolerant symbionts, they have a much higher threshold to heat. Okay. And now we can't forget about our microbiome, right? So our microbiome, our bacteria, influence our corals resistant to disease. So our Acropora cervicornis, our staghorn corals, we know that in Florida, they have two different types of microbiome profiles. So we have our resistant corals here on the left, where they have a much more diverse microbiome, so that they have a lot more different colored bars in there, means they have a lot more bacteria. And then over here, most of our genotypes have a microbiome that's dominated by these bacteria called Acharicotia roweri. I butchered that name. But the Acharicotia, or RICAs for short, they are this obligate parasite. So they steal energy from the coral, and they reduce the coral's fitness and just overall health. And corals that are dominated by this parasite are more susceptible to disease. All right. I want to get into one experiment that we led for disease resistance. So in 2022, in collaboration with several partners, we conducted the largest SCTLD transmission experiment ever. And I'm going to say that in finality there, it was the largest one ever, where we screened nursery beard massive star coral genotypes to identify stony coral tissue loss disease resistance profiles. And we do disease transmission experiments by taking a diseased main fragment and setting the genotypes that we're testing up right next to the disease lesion. So it's like a direct contact transmission. Okay? And then we measure things like whether or not they become diseased or stay healthy, how long did it take for them to have disease signs, and then how quickly that disease progresses. And that gives us kind of the traits of those kind of disease traits, really. Now, this experiment in numbers looked like 3 drive, sleep, dive, drive trips to the Keys from Sarasota to Summerlin to collect our diseased corals for our experiments. 4 experiments over 4 months, which had 50 disease-exposed tanks and then 20 control tanks in the lab, which had up to 270 individual corals in the lab at one time, so we were tracking a lot of corals. And we tested 154 genetic individuals throughout all these experiments. And we collected over 2,000 samples that we sent to our collaborators for determining a biomarker for SCTLD resistance. So this is a lot of work and not a lot of sleep, honestly. So the bad news from this is that we found pretty low levels of resistance. Only 15.1% of the corals that we tested did not develop disease signs. So they all got diseased, right? Now, I use this hierarchical clustering analysis, which is just a fancy way of saying I took all of their different traits we measured, threw them into a model, and it grouped them into different disease resistance groups. So our corals to our red and orange colors were our losers. Those are the ones that were super susceptible to stony coral tissue loss disease. And then our greens and blues were more resistant. So the good news for this experiment was that our results agreed with what we were seeing on the reef. The same genotypes that survived the experiment were also more likely not to get the disease when they were outplanted on the reef. So the science was transferable from the lab to the field. So that's good. It's good when our results repeat themselves. It's good. I know it's not good that all of them were pretty susceptible, but it's good when it matches itself. So our collaborators are now analyzing the over 2,000 samples for host genetics, gene expression, and microbiome. So looking at the entire holobiont traits to figure out what sets apart the resistant individuals, what makes them not get the disease. And then that will allow us to determine a biomarker. So basically just something we can scan and say, we know that this profile makes the coral susceptible to the disease. So that way, all we have to do in the future is grab a small little sample from our corals used in restoration and then run that through some lab tests. And then we can figure out, okay, yes, this one will be resistant to SCTLD or this one's not going to do that great. Okay. Now, I like to add in one bonus part to this restoration cycle, and that's adding in coral first aid. So a lot of our work in Florida Keys lately has been testing different coral disease treatments. And I don't have too much time to go into all of this, but just a little bit of teaser here. So our work is providing tools to managers and practitioners for preventing the loss of investments in coral reef conservation. So imagine if we had a treatment for these diseases, we could go out and just slap that on top of the coral disease lesion. And then we save those larger corals that were already there of like reproductive size and like already building that reef framework, right? So if we have those treatments and can put those to use in like a very actionable way, we can prevent, we can increase our long-term efficacy of our large-scale restoration efforts. So we are working with a team called Ocean Alchemists. This is Mike and Katie. They're the team behind this company. So they are actually, they started this company in their basement, really. It's kind of as their night job. Mike still works at the pharmaceutical company. And what they do is they are making these coral cure ointments, which are customizable, they're biodegradable, and they're capable of carrying a wide range of different ingredients from numerous natural products. So there's nothing super harmful. It's all just natural chemicals that you would find in nature anyway. And then those active ingredients contain antibacterial or antiviral properties. So these ointments come in a lot of pretty colors, and they're not harmful to the environment around them, but they do stop the diseases from progressing. So this is Aaron and I applying the coral cure treatment on top of a large coral at Lou Key. I have named him David, right? So David, I think, is still alive and thriving. I'll hopefully see him this week. And so we are covering black band disease with this ointment. And we can see here we've kind of gone from using syringes and small caulk guns to industrial sausage guns with 3D printed tips that extrude the ointment in the right size for treating the corals. We've been doing this coral testing since I started at Mode, so it's been a lot of trial and error. We've been a lot back and forth. Okay, so you see this is actually Katie from Ocean Alchemist extruding the ointment. She's going to break off that piece and then cover the black band disease entirely. And it has to be that wide because we have to go into the healthy coral tissue a little bit, because that black band disease, it's funny. It's made of this microbial consortium. It likes to just grow over things. It grows very quickly. So you have to make sure you smother it completely. Now, the coral will die underneath that ointment, but it would die completely from that disease if we didn't stop that disease from progressing. So just one of our research projects testing the disease treatments is asking if treating every coral with disease in an approximately 1,200 square foot area, so roughly the size of my 2-bedroom, 1-bath house, does treating all those corals save more living tissue than if we didn't treat anything in that same area? So, let me just go back. So here we have a 3D photogrammetry map of one of those sites where we were testing our treatments at. So it's kind of a bunch of our Orbicella annularis corals there. So did it work? Yes. The answer is yes. The corals in our control plots, which are our gray bars here, lost significantly more tissue than the corals that were treated. So the control corals lost 2 times more tissue than the corals that were treated. So we were able to go on this kind of small scale and save more living coral tissue by using these treatments. I'm going to swing back around to heat stress because it leads nicely into the next part of our restoration cycle. So remember, we were talking about how these sexual recruits did much better in the coral bleaching event. And we are actually seeing that we can put numbers to this, and they're way more likely to survive the bleaching event than the wild colonies, as well as kind of these founding genets that are used in a restoration program as well. Here we can see some elkhorn recruits that were originally outplanted in 2022, and they're still alive and thriving today. And then we've got this staghorn coral that was outplanted in 2020, and it is still alive and thriving today. So they're still growing. Our sexual recruits are doing really well. So that leads us into the sexual reproduction stage of our science-based restoration cycle. Now, this process starts with ensuring that we have access to these sexually mature large corals. So corals can reproduce when they get to about the size of a dinner plate, like my face size, I don't know, about that big. So once they're that big, they can start reproducing. And we keep these large corals, actually most of them are now in our Key Largo nursery. So Sam's in charge of all those. And so we let them grow larger. We're not like fragging these guys out. We want them to get nice and big and happy so that they will reproduce for us. And then we have to bring those corals back into the lab unless we want to spend all night trying to dive in the dark watching spawning, because spawning season only happens at night. Well, corals only spawn at night. And they also spawn after the first full moon in August, which is also not always the calmest time of year, right? So we bring them into the lab. And when corals spawn, they look something like this. If my video is going to play, does this one? Oop, okay. It kind of looks like reverse snowfall, right? So this is a staghorn coral releasing gamete bundles. And so those gamete bundles have both eggs and sperm in them. When they get to the surface, they kind of burst open naturally in the wild, but when we are working in the lab, we actually have to collect those little gamete bundles. So what this looks like in the lab is we've got our corals in our tanks. They release their gamete bundles. You have a bunch of people wearing red lights and bending over tanks for a long time at night. We have to shift our cycles to where we are now night owls working on this. We use red lights because that doesn't mess with the coral's natural circadian rhythm. So it doesn't affect their sense of it's nighttime and I should be spawning. So coral spawning, we've got the corals release their gamete bundles. We then break their gamete bundles apart into their separate eggs and sperm. And since we're collecting from specific known genotypes, we can actually do crosses of set parents. So we know exactly who the moms and the dads are for the crosses that we're making. We then have to rear the larvae. So they stay in this little kind of mixing bowl of coral larvae growing out for, depending on the species, a week or two until they're about ready to settle. They kind of morphose into more of a worm-looking thing, as we'll see here. So they kind of stretch out into a planula, which is like more of a bean-shaped coral, so like little balls. And that planula then goes down, has to find a nice place to settle, and where it'll attach to its plug. Once it's done that, it starts changing shape. Go with that. It starts changing shape, and it will also take up its symbionts and start getting some of its color. So here we see we've got a couple little polyps, and it's getting its coral color. And then we have to grow these little recruits out in the lab and give them all of the love and food that they could ever hope and dream of. I became a first-time coral foster mom this year, where we had extra corals that were being grown out, and I brought them to my lab at City Island, where kind of the mothership of MODA is. And so I had 9,000 coral babies in the lab until I sent some off to live elsewhere. It's quite a process. I keep my interns very busy these days because they get fed every day. So we give them all the love and care that we can to grow them out to be used in restoration down the line. So we can also hold and spawn corals in land-based tanks. So these are spawning-sized corals in systems where instead of having to stay up all night, we can actually change their light cycles so that it becomes dark during our daytime, and we can actually come in, have our coffee, and then the corals start spawning, which works out really well. So by using this combination of corals that are brought in from our nurseries to also having corals that are held on land for this purposes, we can grow and create thousands of new corals each year that have a bunch of new genetics that may make them more resistant to the stressors that they're facing. So, like I said, we can take the eggs and sperm from specific individual genotypes of corals. So we use this to help inform our managed breeding program to increase resiliency. So we can take a parent that's resistant to high water temperatures and cross it with a parent that's resistant to disease. And then the hopes is that their offspring is a whole new generation of resilient corals. So that's kind of where we're going with all of this resilience testing. It feeds into our managed breeding program. One last part of this cycle. It just keeps going. So we also preserve our corals within a living coral biobank. Now, we focus on preserving species and genetic diversity on land. So we hold our corals on land so that we don't lose everything to our variable environment on the reef. And so we do this with live banking of live corals, as well as cryobanking, where you can actually freeze the sperm of corals and then unfreeze it later and use it down the road many years later. So we have MOTE's International Coral Gene Bank, which is in Sarasota, Florida. And we are actually undergoing an expansion right now. All these blocks, this is a whole new building. There's another building behind it. So we have a good deal of space at MOTE's Aquaculture Research Park for this gene bank to exist. It's also nice because it's not on the water, so it's a bit more protected from hurricanes, which we deal with very often, right? So it has a lot of backup power sources as well. So we make a very nice safe place to hold all of these valuable genetics of corals. And so the goal of Moat's International Coral Gene Bank is to provide a safe haven for corals that are on the brink of local and regional extinction. To do that, we have to collect more corals. So we are focusing on all 9 geographic regions and multiple habitats to optimize the diversity of corals that we're holding in the gene bank. So here we have all the different regions, kind of like spatial zones of Florida's coral reef, as well as the Gulf. And then we're collecting from kind of our near shore, our mid-channel, and our offshore habitat zones to capture diversity of genetics, because those corals from those different areas are going to be used to different stressors. So we're hoping to capture resilience by having them from different locations. Okay, so again, this is showing kind of inside our coral gene bank. We have a bunch of these raceways where we're holding corals, and it's kind of fun. We use RFID chips, like if you chip your dog, we put that inside the coral plugs to help us keep track of the corals in the gene bank as well. So you can just take a little scanner and know which genotype that is, just in case our handwriting wears off. So that's fun. So like I said, we also use cryopreservation for reproduction and conservation. So in 2024 was the first use of cryopreserved sperm from a 2023 coral cross with fresh eggs from 2024. So we took parents that spawned in different years and were able to cross them by using cryopreservation. And we created hundreds of new babies that would never have existed without that technology and training. So we also contribute samples from our spawning efforts through cryopreservation to a larger biobank gene bank as well. Now, the current gene bank inventory is 27 species and 4,028 colonies, which is pretty incredible. It's a lot of corals to keep track of on land in Sarasota, Florida. And a lot of these corals are predominantly the staghorn and elkhorn corals at the moment, because we went through and we made sure that we had representatives of pretty much all of the living at the time. So this is before the 2023 bleaching event. These are where all the staghorn and elkhorn corals were along Florida's coral reef that we actually got living collections of that are being held in our gene bank. So 250 staghorn and 150 elkhorn colonies, all represented in Moat's gene bank. There are now only 37 wild elkhorn colonies remaining on Florida's coral reef, and I would say less staghorn corals. So coral restoration and gene banking preserve the genetic diversity of Florida's elkhorn and staghorn corals. Without having gene banking and restoration, already having these corals in kind of propagation, we would no longer have these genetics left, which is very important because having those genetics also increases resilience of future corals, right? We can use those in our spawning. So it's a very important part of our restoration cycle is this gene banking or live biobanking. All right, so I'm just going to do a little teaser here, but we are adding another location to Moat right here at Pinnicamp, and I am not quite the one to talk fully about this, but we are adding the Moat Marine Laboratory Coral Reef Ecosystem Nursery at John Pinnicamp Coral Reef State Park. From what I understand, should be here in a year and a half or so, something like that, but I will let someone else answer questions for that. So this will be focused on really education and kind of outreach efforts, as well as propagating more corals. There will be seagrass growing out here, as well as our grazers. So it's going to be a whole coral reef ecosystem being kind of grown out for restoration at a facility based here at Pinnicamp. So that's pretty exciting. So that's another next step. All right, so so a lot of what I said tonight is pretty depressing. I'm hoping I gave you a little bit of hope. And I want to say that there are a lot of ways that you personally can help with what we're doing on Florida's coral reef. So there's a lot of ways. One is you can get involved and dive. So there's several dive programs with our restoration programs in the Keys. So you can get in the water with us and work in the nursery. You can volunteer with us. So you can volunteer at our land-based nurseries as well. There's a bunch of people on the team here that can talk more about that as well before you head out tonight. You can also just visit. If you want to kind of learn more and actually see these corals and raceways that I talked about tonight, you can visit our Florida Keys land-based nurseries. You can actually do hands-on experience. So if you're like, oh, I really want to fragment a coral, you can do that. So you can get involved and work on that as well. Also, if you are a Florida registered driver, you can get a Protect Our Reefs license plate. And the Protect Our Reefs license plate actually fund our research. I have a POR grant right now that's actually looking at if we can dose the right kind of bacteria onto the mustard hill coral to figure out if that will make it more heat resistant. We have another POR, so the data in here that was looking at the sexual recruit surviving the bleaching event. When we're looking at their microbiome, that data was a part of our project that's funded by Protect Our Reefs license plates. So it's funding research by Moat, but it also is an open grant program for other researchers and restoration practitioners throughout the state as well. So that's one way if you drive, you can get this nice vanity plate. Erin has the coral POR plate. So I'm a little mad that she grabbed that one, but she's always been the coral doctor. She's got that taken, so you can't have coral. Lastly, you can always donate as well. So this QR code will take you to a page where you can donate to our coral work at Moat. And then I kind of want to bring it home to also just what you can do in your daily lives. This is a great graphic that was made by Noah that just kind of shows different kind of values and choices you can make in your daily lives to help kind of conserve our marine ecosystems. And I kind of add two of my own as well. You know, one of those is learning about and engaging with coral reef restoration and conservation. You're doing that tonight, so check. You guys are already helping save corals. And then I want you to go out and share what you've learned tonight with your community. That's a big part of this, right? If we don't know about something, we can't work on protecting it and doing better. So go ahead and share what you've learned tonight with your community. If you've learned anything, I hope you have. So with that, I will take any questions. I think we have a microphone for questions. And if there's no questions, I'll just run out the door. Let's see. Up front, are we passing around no, maybe we're not. Okay, I'll repeat it. There we go.
Any thoughts on why, at least in the Keys, patch reefs are often healthier than the reef track itself, even though it seems counterintuitive in terms of temperature, turbidity, nearshore pollution? I just never been able to figure that out.
Okay, for the folks on Zoom, the question is, any thoughts on why the patch reefs are sometimes more healthier than our reef tract areas? That's a great question. And so most of my diving in the Florida Keys has been in the lower Keys. And a lot at our mid-channel to offshore reefs, some on our near shore. And if we think about maybe our nearshore corals being more healthier, it could be because they're already experiencing those stressors on a daily basis, right? If they're in shallower waters, they're already having a lot more variation in their temperatures and light environments. So they're kind of already predisposed to having to deal with those stressors, so that makes them more resistant possibly. Yeah, so that's part of why when we do collections for gene banking, as well as getting new corals into our restoration, we're focused on getting them from different areas, especially those areas that might be already more resilient as well. Yeah, right.
How do you attach the coral? The organism itself.
Yeah, that's a great question. So the question was, how do we attach the corals to the substrate that we're growing them on without harming the coral? And that's because when we frag them, the bottom bit is just skeleton. So the top is where the living coral tissue is, and then we take the bottom part is a skeleton, and we just glue. Sometimes there's different groups use slightly different methods. Sometimes there's like a two-part epoxy, but you can use glue or epoxy, just glue the skeleton down. And so it's not touching the actual coral tissue. Oh, over here.
Sara worked on trying to determine what genes are responsible for beneficial corals.
Yes, so the question was work being done on which genes are beneficial for corals. So a large part of that disease transmission study is where they've taken the samples to look at gene expression, which will tell us which genes are responsible for the coral's resistance to stony coral tissue loss disease. There's also a paper by Steve Vollmer, who's now at one of the Florida schools, looking at genes responsible for I think it was heat tolerance in the staghorn coral. So there's a lot of different work being done to understand which genes and how they're being expressed are important for coral resistance. And there's a question right behind you as well.
I think it's
Similar.
to what you're saying. I had a possibility of, although it's bad terminology these days, genetically modifying some of these.
Yeah.
Some structures to be able to be more resilient rather than doing selective breeding and finding the pieces that would naturally happen by that process.
Yeah, so that's a great thought as well. So the question was kind of, is there work being done on genetically modifying corals? And we are just at the beginnings of being able to genetically modify corals. I was actually at a workshop hosted by the University of North Carolina, Wilmington back in August of last year that was just about that whole topic. So they have a project there where they are working on trying to genetically modify corals based on a trait that they think is responsible for heat resistance. So you have to like find the trait that you want to actually like put into the corals, and you have to actually get it into the coral. You can only do that as their larvae before they settle. So it has to be done like right after that fertilization process. So it can only be done like once or twice a year, which is really tricky. And then it's also, should we be doing that is always a conversation, the ethics of it. So that was a big part of our workshop, was talking about the ethics of genetically modifying corals and, you know, what's the risks versus, you know, if we don't have these corals in the future, you know, what are the pros and cons there? So there's still a lot of work being done on that and making it possible, as well as should we do it? Yeah. Oh, yes.
So when you say you take it back to the laboratory after you sniff it or whatever, are you close here? Or you take it back to Sarasota, or are you still lab here?
Both.
Time frame? I mean, how much time?
Yeah, so great question. So the question is, you know, as we're fragmenting corals maybe from the reef and bring them back for experiments or for growing out, how quickly we have to do that. And we try to be as quick as possible. We, like on the boat, we'll keep them in buckets with water, or just corals actually do really well transported in wet bubble wrap. You put them in a cooler to keep them just kind of like temperature fairly constant, and then you just put wet bubble wrap on top of them. You kind of keep them moist, and they're okay like that for, you know, 12 hours or so. You want to like refresh the water. I will not write a protocol for that, but that's how we typically do it. So like I said, when we had to collect the disease corals for our experiments, we were, you know, driving down here, sleeping the night, diving, collecting the corals, putting them in a cooler, driving right back up to Sarasota. So that's just like a 7-hour drive. So they're fine in those coolers for about 8 hours. We put them directly into water afterwards. So we have a lab up in Sarasota, and we also have the lab down in Summerland Key, where we also do a lot of our experiments. Yep. So they're fine for a short time. Yeah.
Are you other parts of the world, of course, they're doing research, are they having similar results to their research as well?
Yes. So the question is if there's similar results across the world in different coral research groups. And the fun thing about coral science is it's a very small world. You get to know everybody. It's a lot of people, but everyone knows each other at some point. And you know, a lot of work that we hear about in the news media in particular is about the work being done in the Great Barrier Reef. They have different species that they're working with. So sometimes the results are slightly different than ours. They have a lot more diversity as well, so some of it's a bit different, but we are able to learn from their research and bring that into our projects as well. And it's really a constant conversation of all working together to help, you know, build back our reefs worldwide. So similar results. I can't pinpoint any specific studies, but yes.
Possible that there's some species from transplant here.
Well, physically possible. Should we do it is another question. So the question is like, if it's possible to bring coral species from, you know, different parts of the world to here. And that's, we wouldn't want to do Australia. We're having a hard enough getting corals from the Caribbean from a different space over to here in the Florida coral reef. And that's all, you know, and like for good reasons. We have to make sure that we're not going to bring with that coral from like Australia over here, like a different bacteria that's then going to cause some outbreak of disease here. Yeah, so, yeah. Oh, no, it was a great question. And these are like conversations that we have on a monthly basis almost in the coral world. Like so recently we have this large collaboration called the Reef5 collaboration, and it's with I think like 10 different partners in the Florida, including the University of Miami. That's who it's led by. And you might have seen something in the news about the Flanduran corals, and those are corals that were, so it's elkhorn coral from Honduras crossed with Florida elkhorn coral. And they actually are getting permits to be able to outplant those corals on Florida. So they were raised in their labs at, I think, the Florida Aquarium and the University of Miami. So like crossing those, because those Honduran corals actually are more resistant to thermal stress, or we think they are. So conversations are starting. It's a good question. It's like constant conversation. Yeah. Let's see here.
Any other questions? Well, thank you very much.
Thank you for your presentation and for sharing your wealth of knowledge with us. And thank you to Moat for sponsoring this program and for our friends to help us in having it here. Like with the questions and gift bags. But before you leave, I'd like to give everybody some important information. As was mentioned by Anne, this series was started actually a long time ago in 1992 because